so this one, just like the last one, what do we want to do with it? The first thing that we want to do is we want to take the text, convert it from that story problem form into something that is more manageable for us. So always the first thing that you do is you say, okay, what is dominant, what is recessive? Make a little table. So here I've already done that. White is dominant from last time, yellow is recessive from the last problem that we did. Disk shape is dominant to sphere, little s for sphere, big S for disk. Okay, so we have that set. Now what are we going to do? We're going to convert what it's telling us up there in text. So it says heterozygous for disk, heterozygous for white, right? We're going to draw out that genotype. So heterozygous for disk is going to be big S, little s, right? Heterozygous for white is going to be big Y, little y. Okay, now we have the genotype of the first individual in the cross. The second individual in the cross is yellow sphere shape. So it doesn't say heterozygous. Why? Because in order to be yellow sphere, it has to be homozygous, right? So yellow, we cross it, we make a little x, that's telling us that's the one that we're crossing. Little y, little y, little s, little s. Okay? So from this point, right, it wants us to say, okay, what is the phenotype ratios for the F1 generation? So here we can do a Punnett square. Because this individual, the recessive phenotypic individual, is only going to produce one gamete, right? The only possible gamete it can produce is little y, little s. It's not going to be as big of a Punnett square. You only have you could draw out 4 of these, but it'd be sort of redundant, right? Here, what are the genotypes here? Did anybody get this far? What are the possible genotypes? I mean, not genotypes. What are the possible gametes?
Big Y, big S.
Okay, here's one. Big Y, big S. Good.
Little Y, big S.
Little y, big S. Good. What's another one? Big Y, little s. How about that one? Big Y, little s. And then little y, little s. Okay, so now we could actually do a Punnett square where it's just going to be like this, right? Since there's not room up top, I'm going to put this one on the bottom. Little y, little s. Normally we'd have that on the top. It's only going to be a 4-celled Punnett square, right? So you're going to have, what's this genotype going to be? So big Y, big S, and then bringing this one up, what is that going to make it? Big Y, big S, little s. All right? So what is that phenotype? And this is all really good practice just in the beginning. What is this phenotype going to be? So it has the big Y, and it has the S, big S, right? It has that dominant. It's going to be white disk. All right, we got one phenotypic class right there. Now what's this one going to be? Little y, big S, little s. Does everybody see how I'm combining those to get that in the Punnett square? So what is that genotype going to be, or phenotype? Yellow, right? Little y is yellow, and big S, little s is what? Disk. All right, here's that second phenotypic class. This one now, big Y, little s, right? That's going to be white. What's the shape going to be? Little s, sphere. Okay, sphere. And this one is going to be little y. That's the recessive phenotype. Little s. That's going to be then yellow, yellow, and then sphere. Okay, so now the ratio is, when we did the cross for the 2 heterozygous individuals that were heterozygous at both of those, we got the 9:3:3:1 ratio. Here's another ratio you need to remember. This is a 1:1:1:1:1 ratio. This is something we call a test cross. So a test cross, and we'll get a little bit more into it later, but basically a test cross is telling us that if we didn't know, if we just knew the phenotypes of these two, and so this one individual was white and disc shaped, we didn't know what the other allele is, we could test it by crossing this individual with a recessive individual. Okay? If this one was big Y, big S, big S, what would our ratios be for the next generation? Can you do that one in your head? If not now, you will be able to. What's the what if it was big Y, big S, what is the gamete that that individual would produce? Anybody know? Yeah, so the gamete, it would just produce a big Y and a big S gamete, right? Big Y, big S gamete. If this was homozygous big Y, homozygous big S, what's the gamete that this one's going to produce? Little y, little s, right? Combining those two together, you get big Y, big S, little s. All of your offspring would be dominant phenotype. You therefore know, you can therefore, because of that result, you can say, okay, we're quote unquote testing what this other allele is. Okay? If in fact you get a 1 to 1 to 1 to 1 ratio, what does that tell us? Yeah, exactly. That this is big Y, big S, little s. That's why we call it a test cross. Okay, so I encourage you again to just keep practicing these, keep going through these. Eventually, if it's a little confusing now, the more that you do it, you're going to get so good that you'll be able to